As reported by the World Health Organization and Alzheimer's Disease International, in every 3 seconds there is one person diagnosed with Alzheimer's disease. As the aging population is expanding, dementia has become a tremendous socioeconomic issue. At the moment, there is no cure for AD. The current medical treatment for neurodegenerative diseases could only help to relieve symptoms. How can we detect or even treat neurodegenerative diseases with current technologies? Through this video, you will learn how the HKBU School of Chinese Medicine and the Faculty of Science could bring hope to the public through their research results. AD is an irreversible, degenerative brain disease that affects brain functions including memory, thinking, behavior, and the ability to perform daily activities. Although AD is incurable at the moment, early interventions and timely treatment could help the patients to retard the progression of the disease and to prolong their normal lives. The currently available method for diagnosing Alzheimer's disease is to extract cerebrospinal fluid from the lumbar spine. The extracted biomarkers could then be tested in the laboratory. The patient experience of this invasive detection method is painful, following with undesirable side effects. Usually, as the patient develops the symptoms and go for diagnosis by CSF, they are at the middle or advanced stage of the disease. To address the issue, Professor Ricky Wong and Professor Li Hangwing from the Department of Chemistry of Hong Kong Baptist University led their research team to invent a new AD diagnostic kit.
We have invented a new AD diagnostic kit that tests blood, saliva, or urine, providing a faster and more sensitive, affordable, and less invasive way than traditional method of detection.
Our detection assay only requires a few microliters of the body fluids to quantify the target protein biomarkers. Once the AD biomarkers is captured by the magnetic nanoparticles, an immunocomposite is formed. Then, the nanocomposite will be labeled by newly developed silane compounds and gives fluorescent signal for quantification. The intensity of fluorescent signal acts as an index to evaluate the severity of AD.
When compared to commercially available ELISA kit, the new diagnostic kit provides an option for the patients and allows AD diagnosis using their saliva and urine. In addition, the concentration requirement of AD biomarkers being detected for the new kit is much lower. Besides, our new kit takes only 1.5 hours to obtain result, while the ELISA kit may generally take 18 hours. For a typical analysis, speed is one of the merits. It saves running cost. Short time allows future point-of-care products more feasible as well. Last but not least, the cost of the new kit is about $100, which is 50 times cheaper than the ELISA kit.
With this kit, we hope to lower the barriers for early-stage diagnosis of AD in order to help the elderly maintaining better quality of life by timely intervention.
Neurodegenerative diseases are incurable at this moment. The currently available treatments are mainly for relief of disease symptoms but not a cure. Therefore, replacing dying neurons by neural stem cells can be a new and effective cure. In Hong Kong, the scientists of Hong Kong Baptist University developed a potential personalized stem cell treatment by using the neural stem cells obtained from the patients themselves for their own treatment.
Actually, in the present research, we have employed nanoparticles, and these nanoparticles are magnetic nanoparticles. We use this characteristic of the nanoparticles to harvest the stem cell. We can apply magnetic field, extract the nanoparticle tag cells to migrate, and then we can harvest them. So this is a kind of a new application in the adult brain. We are among the first groups to do that.
Then we can produce the stem cells outside the body. HKBU research team from the Department of Physics and Biology have invented inorganic sculptured extracellular nanomatrices to avoid using the animal or any other biochemical growth factors for cultivating cells.
Physiological and chemical stimulation has been widely used to control cell fate. In my lab, we use glassine angle deposition to fabricate the nanomatrix. So this is our product. We can flexibly control the structure of the nanomatrix. And after that, we put the neuron stem cells onto the substrate. And the neuron stem cells will assemble it as the neuron sphere. And after that, this neuron sphere will differentiate into the functional neurons.
Using this technology, the cell growth rate is more or less than the same as the current methods with growth factors. Importantly, this technology can address the safety issue. This technology is feasible to be applied on human because the material is biocompatible with no toxicity. The stem cell differentiation will be operated in vitro.
We will seek the clearance from the European agent, that's the CE mark, and also FDA from the United States. After we can get the clearance, we can then market our product to the clinics.
Traditional Chinese medicine has a long history and there are so many prescriptions and applications. TCM has been familiar to people. For example, Oncaria rhincophylla is a common herb used for the treatment of hypertension and Parkinson's disease. Professor Li Min from the School of Chinese Medicine of Hong Kong Baptist University has nearly 30 years of clinical experience. As a natural herb, Oncaria rhincophylla is safe to use. Her research team conducted an in-depth investigation of it for the treatment of Parkinson's disease. The new drugs derived from the herb brings hope to patients.
I found that Oncaria rhincophylla is very effective in treating Parkinson's disease, especially in improving the symptoms. Based on my clinical experiences, as well as a large quantity of literature reports for these herbs, our team studied its active ingredients and pharmacological effects. The main bioactive ingredients in Oncaria rhincophylla are coranoxin B and coranoxin. These two bioactive monomers are efficient in the removal of alpha-synuclein protein, which is the major cause of Parkinson's disease. Oncaria rhincophylla can induce autophagy in neural cells and thus promote the clearance of alpha-synuclein protein aggregates. This finding had never been reported nor discovered.
Professor Li Ming's research pushes back the boundaries of traditional theory. Professor Li does not only focus on bioactive compounds in herbs, but also conduct in-depth research on traditional Chinese medicine formulations and made a breakthrough.
We found that the modified Huangnian Jiedu Tang promotes the degradation of amyloid beta peptide and tau aggregates for both in vitro and in vivo tests. It can also pass through the blood-brain barrier and clear the accumulation of these proteins, which is the major root cause of Alzheimer's disease. Even though the mechanisms in Parkinson's and Alzheimer's disease are not the same, we shall modify different bioactive ingredients to enhance their pharmacological and bioactivities. A new drug for Parkinson's disease can then be developed by combining different bioactive ingredients of traditional Chinese herbs. This is a part of our plan.
HKBU researchers hope to invent new and better treatments for different types of neurodegenerative diseases through various efforts in order to help more elderly people get timely treatment through testing and to maintain their normal quality of life.